this is the synopsis of the course. It's fundamental microbiology. That basically means you're learning everything about the fundamentals of the science microbiology. So you'll be introduced to concepts and basic techniques of microbiology, evolution, structure and function of prokaryote, dissemination, control and identification of microorganisms, exposure to aseptic laboratory techniques for transfer, isolation and staining of microorganisms. Okay, so microbiology is about microbes, right? So when you want to know, microbes are everywhere, yes? That means when you are handling microbes, you have to be especially careful, because if you're not careful, your results will be you'll get contamination. Okay, so an essential part of this course is to teach you aseptic techniques. Okay, the aseptic techniques. Aseptic techniques means where you are able to handle the microbes without causing contamination. Among the aseptic techniques is in the transfer, isolation and staining of microorganisms, which means when you transfer the microorganisms from your plate to your broth to your slides. Okay? You need staining because staining is the essential first step in identification. So the course outcomes are as above, to distinguish the various types of microorganisms. So when we talk about microorganisms, we do not talk about bacteria alone. We talk about bacteria, protozoa, algae, phytoplankton, viruses. Okay? Everything that can be considered microbes. Then to understand the core concepts of microbiology, then to demonstrate basic practical microbiology skills. So what is microbiology? A study of small living things, usually too small to be visible to the naked eye. So the microscopic organisms can include bacteria, archaea. Archaea is also considered a prokaryote. Then we have viruses, fungi, protozoa, and algae. Microbiology is an important science. It's the small microbes that are extremely important in the Earth's ecosystem, in the Earth's functioning. Their importance is in the form of nutrient and energy flow. The bacteria recycles, right? The role of the bacteria is to regenerate, recycle, decompose. Okay, so without bacteria, none of the elements can be recycled back. Okay? So that's why, if you remember, nutrients are in cycles. Without bacteria, the cycle doesn't flow. Okay? So that means bacteria plays an essential part in the nutrient and energy flow, plays important roles in decomposition, also in the production of food, drugs and vaccines, in the form of fermentation, in the form of probiotics, okay, in the form of yogurt, right? All this. For drugs and vaccines, usually is the use of microbes as a host to carry a recombinant DNA to produce the vaccines and the drugs. Okay. Because it is cheaper to produce drugs, certain chemicals using bacteria as opposed to chemical synthesis. Okay, because you can carry out fermentation to produce a large amount of the metabolites that you require. Okay, so when you apply microbes in these industries, it's called a form of biotechnology. So when you apply biology to produce useful stuff, then it is called biotechnology. So bioremediation is important because without bacteria that carries out decomposition, a lot of the waste that we produce will accumulate and make Earth inhospitable. We can no longer live there. Okay? That's why the role of bacteria is important. Now you know about the problems with plastics, right? We have a lot of plastics in the form of microplastics, nanoplastics. So plastics are found almost everywhere. And most of you probably have plastics in your body. Recent research shows that even your lungs and your nasal area, scientists have been able to find strands of plastics, fiber. And it will take time before we realize the consequences of plastics upon our health. But among all the organisms in this world, there is bacteria that can degrade plastics. Okay? Discovered by the Japanese, Legionella sakazaki. So they have found bacteria that can degrade plastics. So there are research carrying out all over the world trying to optimize or try to extract the enzymes or genes that carry out this process in the hope that you can apply it and then reduce the plastic contamination. And of course, infectious diseases, anything that is not natural on us has the potential to cause disease. Anything that is even naturally on your body can cause disease if given the opportunity. Agree? The only reason why we are not sick, even though we know there are lots of bacteria on us, is because we have defense system, we have barrier. The skin is one of the best barrier against infection. Well, that's why if you have a deep cut, then you have to take extra precaution. Deep cut is where you can see the tissues, not a superficial cut. Okay, so there's a difference. If you have deep cuts, you cannot go swimming, you cannot go to the seas. Okay, there are bacteria that can cause necrotic diseases. Necrotic diseases means the bacteria will eat your tissue. So we have immune system, we have our defense, but there is always a chance that it can be compromised. Microbiology is one of the largest, most complex of biological sciences because there's so many things you can study. You can study bacteria, protozoa, helminths. Helminths are like worms. Phycology is algae, phytoplankton. Mycology is fungus. Virus, virology, parasitology, immunology, and molecular biology. And environmental microbiology. So such a diverse field. Here is written basic and applied. Basic pure science and applied. So you have basic pure microbiology where you study the taxonomy, the genetics, metabolism, and the immunology, epidemiology, and etiology related to disease. But whenever you apply knowledge, then it becomes applied science. You can find the application in environmental microbiology, as I mentioned, food and beverage, pharmaceutical, genetic engineering for vaccine production, okay, and infection control and chemotherapy. Microbiology is very, very important in the beginning of molecular biology because the primary organism that they used was E. coli, Escherichia coli. It grows fast and it's easy to handle. So from there they are able to understand a lot of the biochemistry and a lot of the genetic processes. And because there is similarity in the processes among all organisms, they are able to translate the understanding faster. Okay? That's why E. coli is one of they call it a laboratory pet because it's so easy to grow, so easy to handle. Origin of microorganisms, 3.5 billion years ago. The belief is the simple organisms emerge first. Okay, simple organisms, which means the single cellular prokaryotes, they emerge first before it become more and more complex. From prokaryotes, you have single cellular eukaryotes. The difference is you have an envelope nucleus, and then you start seeing organelles. Okay, and then later on the eukaryotic cells continue forming to become multicellular. When you talk about the evolution of microorganisms, there are essentially two types of classification. The traditional Whittaker system of classification, where a lot of things are done visually. So the classification was visually. You see animals that they look alike, they group as one. That's why you have the green plants on one side, you have fungi on the other side, and animals. Okay, so it's more of a visual, and certain characteristics they are able to differentiate them into groups. But now we have the taxonomic classification based on the ribosomal RNA gene. The gene of interest is 16S rRNA for prokaryotes and 18S rRNA for eukaryotes. What they do is they extract the DNA from the organism, they amplify only the gene of interest, which is the 16S or the 18S, and then they send for sequencing. Then from the sequence, they are able to carry out alignment to see what species it is. Okay? So most of the time, once you get the sequence out, you have to use the NCBI gene bank to identify the sequence that you have. Okay? From the 16S, they discovered that there are three essential domains: bacteria, archaea, and eukarya. So eukarya are those with 18S. These two are the 16S. The use of ribosomal RNA sequences in identification was first postulated by Carl Woese. Okay. So he's the one that came up with this and started the initial database for identification and taxonomy. So if you notice, then the identification system depends on the database. If the similarity is 99% and above, you can accept it as a similar species. But if it's between 95% and 99%, then it could be the same genus, but maybe a different species. Then you have to do more work. You have to do other tests and do alignment with different species within the same genus to see which one it is most closely aligned to. Okay? So that is in the field of bacterial taxonomy. In bacterial taxonomy, that's what you will have to do. So now essentially we'll look at the general differences between the prokaryotes and the eukaryotes. As I've mentioned, prokaryotic cells are very simple. You have a cell wall, you have the cytoplasmic membrane, cytoplasm, then you have your DNA. Sometimes the bacteria will carry plasmids. Plasmids are considered extra chromosomal elements, which means extra chromosomal means it's apart from the chromosome. Okay. So plasmids are important because most of the time they carry antibiotic resistant genes. Okay, so that's why, and these plasmids are able to be transferred among the bacteria. Not only same species, but even different species. And that is why the world has a problem with antibiotic resistant bacteria now. All the antibiotics in this world, there is a bacteria that is resistant to it. It's just whether the resistance is common, predominant, or rare. Then we have eukaryotic cells is where you have organelles, okay, enclosed organelles with different functions, and also the nucleus. So here you can see the definition of unicellular, consisting only single cell, multicellular, consisting of numerous cells. Okay, so this is the differences, general differences between prokaryotes and eukaryotes. First, eukaryotes are larger. Okay, eukaryotes are larger, and eukaryotes have true nucleus. Then they have organelles, and usually for cell wall, it's more prominent in prokaryotes. Here you can look at the ribosomes. So ribosomes are composed of a large subunit and a small subunit. So 50S and 30S. So in prokaryote is 70S ribosome, which is made up of the 50S large subunit and the 30S small subunit. Same goes for the eukaryote. The 80S ribosome is made up of 60S and 40S subunit. So the S is a Svedberg unit. So within the large subunit, you have this 23S and 5S ribosomal RNA. Then within the small subunit, you have your 16S rRNA. Okay. So 16S rRNA is the standard sequence that we use for identification. So why don't we use 23S? Why don't we use 5S? 5S is too small, and 23S is too big. So we use one that is just good enough to provide enough information for identification. A 16S ribosomal RNA gene sequence has about 1542 base pair. So for prokaryotes, the small subunit 18S ribosomal RNA is used mostly for identification. Okay. But in eukaryote, it's a bit more complex because sometimes they use the ITS, intertranscript spacer area. Okay. So in chromosomes, you have single circular chromosome, here multiple, with histones, and it lacks histone here. Okay. So these are some of the differences between eukaryote and prokaryote. From macroscopic, microscopic, ultra microscopic, and atomic. Okay. Anything that is microscopic is you can no longer see with your naked eye. The role of microbes in energy and nutrient flow. Microbes are small, right? But if you estimate the total microbial biomass in the world, then you realize that it is actually the largest component. But if I were to ask you what is the most abundant, most numerous organism in the world, viruses, right? In the Earth's ecosystem, okay, if you think of Earth as a solitary unit, whatever that's been produced has to be recycled, right? But if there's no external source of energy, then it's static. You cannot grow. You cannot increase in population. You cannot increase in numbers because there's no extra. So sunlight is the only extra energy that the Earth obtains. And the only organism that can trap sunlight are the phototrophs, the primary producers. So that's why they are very important. And among the phototrophs, you have algae, you have, of course, you have large plants, but you have a lot of algae in the sea, in the lakes. Okay? They also perform essential photosynthesis to trap the light energy and produce food, produce sugars. Okay? So without photosynthesis, there is no food. Okay? Only after that, then you have your herbivores, you have your carnivores, and then because there are primary producers, it is able to sustain the food web. Okay? So within the photosynthesis also we have microorganisms that play this role. And we also have microorganisms that break down break down organic matter so that the nutrients can be recycled back. Okay, here you see the common characteristics of the different microbes. For bacteria, unicellular, you have three major shapes: Bacillus, Coccus, Spiral. Okay. These are the major ones. So Bacillus is rod-like, Coccus is spherical. Spiral, it can be curved or helical. Bacteria has a cell wall that is made up of peptidoglycan, and it may possess flagella. For fungi, they are known as decomposers, known as saprotrophs. They have thick cell wall. Chitin is important. Chitin can be used to characterize fungi, which means if you want to know how much fungi is in the environment, if you do not want to culture them or isolate them, then you can actually test for chitin. How much chitin is in that environment? So fungi plays an important role because it decomposes wood much better than bacteria. Okay. Then algae, the one that important because they carry out photosynthesis. Archaea is the other domain. Archaea is interesting, because in the beginning they are considered extremophiles because they are always found in extreme environments. Example, deep sea, easily more than 2,000 or 3,000 meters. You can find them in the deep sea hydrothermal vents. You know that the Earth is constantly moving, the geological plates, right? That's why we have earthquakes, because the plates are moving. So on these active fault lines, there is an area where the new Earth is emerging. Okay? So when the new Earth is emerging, we have lava from the magma when it comes out. So it supplies a lot of new elements and heat, and only bacteria, only archaea can colonize these habitats. So these are deep sea hydrothermal vents. It is very interesting because this is the only habitat that doesn't require sunlight, which means this ecosystem can survive without the sun. All other ecosystems on Earth requires sunlight except the deep sea hydrothermal vents. They are able to sustain itself from the warmth of the magma and the nutrients from the earth. They are mostly chemoautotrophs, which means they are able to produce food from chemicals, not photoautotroph, but chemoautotroph. You also have next protozoa. Their role is mainly in the environment, in ecology. The role of protozoa is to actually control bacterial population. Viruses are here. So viruses are interesting because they are obligate intracellular parasites. That means they will not survive without hosts. So how did microbiology came about. So in the past, like they have this spontaneous generation or abiogenesis, because when they look at something spoiling, when they see something, a fungus or something growing on some, like if you have a floor, then you see suddenly algae or moss growing on it, then they think it's spontaneous generation. So this was like before the 1700s, and there wasn't any microscope also for people to see the microbes, so that they don't consider that it could have been transferred from other places, but they thought that it's spontaneously generated. But because of spontaneous generation, there cannot be any progress in science, because whenever you get sick, they said you did something wrong. That's why you are sick. They don't believe in diseases. They don't believe in infectious organisms. Okay. The most important is they didn't know that there was organisms that can infect and cause diseases. So, among the important experiments that disputed spontaneous generation, first is by Francesco Redi from Italy. He carried out this simple experiment. He placed meat. One is not covered, one is covered, and the other with a gauze or cloth netting. So after a few days, the one with the open jar, a lot of flies and maggots. The one that is closed, nothing. And the one here, he saw maggots here, but nothing on the meat. So he concluded that there is no spontaneous generation. Life is necessary to produce life. So that's the first experiment. Louis Germ, from French scientist, also did something similar with broth. One covered and one not covered, and the one that is uncovered have heavy microbial growth. The one that covered remains clear. These two experiments tried to dispute spontaneous generation, but then you also have John Needham that carried out an experiment where he boiled it, then he covered it up, but there's still growth. Maybe life had been created from non-life, because both heated and unheated test containers grew with microbes. There are also spores. Okay, he doesn't know the concept of spores, and also the boiling, the heating part was not, the duration was not adequate to kill all the microbes. Okay. So that's why his results were not good. Okay, but the dispute for both the Redi experiment and Jablot experiment was that people who supported spontaneous generation said, of course it doesn't grow. There's no air. You enclose it. They said air is essential for life. So you still have two camps strongly against each other. You see these experiments that say there is no spontaneous generation, but then the experiment was not done convincingly, because they said there wasn't any air. How can anything grow? So that was the problem, and it was finally resolved by Louis Pasteur with his swan neck vessel. Okay, so very, very clever design. So he boiled it. It is clever in the sense that it allows air to enter, but because it's swan neck, whatever dust or particle is trapped here and doesn't go all the way in. And that's why the solution remains sterile or clear even after many, many months. Okay? This experiment conclusively debunks the theory of spontaneous generation. So that's why this is a very important experiment for microbiologists. Because if people keep thinking that of spontaneous generation, science cannot proceed, science cannot move on. Okay? And also, other than Louis Pasteur, Anthony van Leeuwenhoek, and Robert Hooke, their microscopes are important because they allow people to see microbes for the first time. Without microscopes, you do not appreciate the microbial world. Okay? So all these samples are still in museums, I think. And in the Pasteur Institute, they have this swan neck vessel, still clear after so many years, in Pasteur Institute in France. From the history, you know that scientists are very stubborn people. So it's very hard to convince them. You need to have experiments, and the experiments have to be done properly. So if you, like for example, if you are a scientist, first you have to observe the natural phenomenon. You see something. Okay? Like for example, if you go to the lake and say, why is it so green today? Okay, you observe. Then you came up with a question, why is it so green today? Because you have a question, you have to have a hypothesis, right? What is the definition of a hypothesis? A hypothesis has to be testable. Because once it's testable, you can design experiments to test it out. Then once you have designed the experiment, you carry it out, then you analyze the results, then you come to your conclusion. So that is the scientific approach. Then whatever hypothesis that you postulated can become theory when it's proven and accepted. Okay? But it takes time. So a theory is a collection of statements, propositions or concepts that explain and accounts for a natural event.
Dr. Jenner was an expert on cuckoos. He was also a good scientist because he asked the right questions. What he really wanted was to conquer the deadly smallpox. Cows sometimes caught cowpox, and milkmaids often caught cowpox from the cows. Cowpox didn't do the milkmaids much harm. What interested Jenner was that they never seemed to catch the deadly smallpox. Thousands used to die from smallpox. Why not milkmaids? Could it be that cowpox got in the way? Could catching cowpox stop them catching smallpox? To test his theory, he needed someone young who hadn't had either cowpox or smallpox. He chose James Phipps, aged 8 3/4. He told the lad he thought he could stop him from ever catching smallpox and that it wouldn't hurt, much. Then he took some pus from Sarah's cowpox and rubbed it into 2 small scratches on the boy's arm. Cowpox was called vaccinia, so Jenner called his invention vaccination. In a few days, James was poorly with cowpox, but he soon got better. 6 weeks later, Jenner took pus from a smallpox victim and deliberately tried to give James Phipps the deadly disease. There was no cure for smallpox, but James didn't get smallpox. He lived to a ripe old age, and now smallpox has been wiped out thanks to Edward Jenner and his vaccination.
This was in the 1700s. Well, a very different time from us now. But it's very interesting. He sees something, he observes, and he thinks. He thinks, why? Why is it like that? Then he proposed an experiment. And he carried out the experiment, and from the results, he concluded, yes, it's true. He might not understand the molecular basis or immunological basis of the vaccination, how it helped. But it helped. And because of that, death from smallpox decreased tremendously. It saved probably millions of people. So you can see here, once the WHO initiated the vaccine program, it essentially eradicated smallpox. But of course, we still have reports of smallpox in isolated clusters because some communities, they still weren't vaccinated against smallpox. So vaccination now is not only against smallpox, right? You have your hip, Haemophilus influenza B, you have your DPT, diphtheria, pertussis, and tetanus shots, right? But all these shots were done before you were 1 year old. So these vaccines are important. These vaccines help prevent death. So vaccines and antibiotics are important. The experiment by Needham maybe is spoiled by the spores germinating. That's why John Tyndall, he carried out successive boiling, which means he boiled something, then he boiled again and again and again, Tyndallization, in order to fully sterilize it. Ferdinand Kohn, he observed heat-resistant forms of bacteria, the endospores from this bacteria. Then this John Tyndall and Ferdinand Kohn, for the discovery of spores and sterilization. Then development of aseptic techniques by these people, Samuel Weiss, Oliver Wendell Holmes, and Joseph Lister. Again, the development of aseptic techniques came from observation. First, they observed a problem. A lot of mothers that give birth in hospitals were not making it. They died in childbirth. Okay, because of infections. So from the observation, they realized that it's because the physicians, the doctors, they were not cleaning themselves. They didn't wash their hands properly. If they are doing their clinical rounds, visiting the sick people, and then they go to the maternity ward, they didn't wash themselves properly. So they were bringing the virus, bacteria, diseases across the wards. So from the observation, they proposed steps to see whether it works. And it does. When they recommended washing, the death rate decreased. And because of that, you adopt it. This is part of the aseptic techniques where you have to make sure there is no cross-contamination. Discovery of pathogens and germ theory of disease. Once you know there's no spontaneous generation, you know there's bacteria. Okay? So the main pioneers are Louis Pasteur and Robert Koch. Okay. So Robert Koch is German, Louis Pasteur is French. So for Robert Koch, he's also very important in microbiology. Later on you'll see that most of the techniques in microbiology, culturing microbiology and all, are adopted from his lab. Okay, Robert Koch. So he came up with Koch postulate. At that time, they have debunked, disproved spontaneous generation. They know there are bacteria, and they know that this can cause diseases. So they were very active in looking for agents of infectious agents of diseases. But to prove that the bacteria actually cause the disease, Robert Koch came up with Koch postulates. So said these are the criteria that has to be fulfilled before we can say that this bacteria causes this disease. Okay? The first is the suspected pathogen must be present in all cases of the disease and absent from healthy animals. So you sample the healthy, you sample the ones that are sick. The ones that are sick must have the same pathogen, the same bacteria. The ones that are healthy shouldn't have the bacteria. Okay, so that's the first rule. Then the suspected pathogen must be grown in pure culture. So whatever bacteria that they obtain, they must be able to grow on the agar plate, make sure it's pure. And then from the pure culture of suspected pathogen must cause disease in a healthy animal. So they use the bacteria, inoculate into the animal, a healthy animal, and the animal must show the same symptoms of the disease. And then the final step is you must be able to re-isolate the pathogen. Okay. So only when you fulfill the Koch postulate can you say that this bacteria causes this disease. Okay. So these are important steps. But this was in the 1800s. Okay? But now we have diseases that you cannot do like this, because we have diseases that only infect humans, for example HIV. So there is no way you can re-inoculate into a healthy person and expect to carry out these experiments, because there's no cure. Okay? So nowadays, for some diseases, there is some leeway, which means they are not so strict. But they still require you to culture, maybe in a tissue culture. Instead of animal culture, tissue culture. Because some diseases, there is no animal model. The disease only manifests itself in humans. But since there's no animal model, you have to maybe use tissue culture or something else. Okay. So now the concept of pure culture, the concept of media, okay, and the concept of petri dish, these are all from Richard Petrie, Walter Hayes, and Koch. So they are all from Robert Koch's lab. Okay, they worked in his lab. So at that time, when they were just beginning, they were using gelatin to make the plate, gelatin to make the agar, to make the solid media. But gelatin melts at a certain temperature, about 37. So it's not. But most diseases are around that 37 degrees Celsius. So it's not suitable. Okay, so they were having problems with that solid surface media. And then Walter Hayes, I think it was his wife, because his wife makes jam. Then he found out that why not use agar also? And that's where the use of agar to make solid media. Because agar is hardier and doesn't melt at 37 degrees Celsius. So they were able to do a better solid media. Okay. Then Richard Petrie also worked in Robert Koch's lab, and he's the one that prepared the Petri dish. He designed the Petri dish as a standard tool in 1887. We still use it now. Okay, we still use it now. But in the past it was glass. Now we use disposable plastics.